Now we study another important mechanism of epigenetic modification using histone modification. DNA is a highly negatively charged polymer. That's why to neutralize those negative charges, DNA is complexed with a positively charged protein, so-called histone. Histone is composed of 8 subunits, and each 2 units of H2A, H2B, and H3 and H4. And they make one complex, and then DNA is wrapping around this histone structure. So this one unit of a globular shape we call nucleosome. Histone is a protein. That's why histone modification is kind of post-translational modifications. Histone can be modified by acetylation or methylation or phosphorylation and ubiquitinations. In nucleosome, 8 subunits of histone proteins are complex like this format, and then DNA is wrapping around the histone complex. Depending on the positions of modification, we can make a map like this. So the first one is H2A and H2B, H3, and H4. Each histone proteins are modified by acetylation and methylation, phosphorylation, and ubiquitinations. Depending on the histone modifications, the gene expression can be activated or can be suppressed. Let's take the example of acetylation of a histone. If the histone is over-acetylated, the chromatin structure is becoming loose, and then this structure we call euchromatin, and euchromatin is a decondensed form. That's why this is active for gene expressions. If this acetylation is removed, then the chromatin becomes condensed, and then this condensed aggregated form of chromatin we call heterochromatin. And the enzyme for this acetylation we call histone acetyltransferase, so HAT. Opposite side of removal of this acetylation is done by HDAC, histone deacetylase. So among different histone modifications, acetylation's function is quite clear. More acetylated, more gene expression. So that's why HAT is activating the gene expression, and HDAC is deactivating the gene expression. That's why HAT and HDAC's function, if they are balanced out, the amount of acetylation on histone is determined. If everything is balanced, then cell is in healthy status. But let's say if the balance is broken, HAT is reduced and HDAC is increased, then as an end result, the acetylation amount is decreased and the more genes are condensed form, and then the gene expression is suppressed. This observation is found in some of those tumor cells. In this case, the suppressed genes may be tumor suppressed genes. To recover this status, maybe we can add HDAC inhibitor, then HDAC inhibitor to block those deacetylation, and then some of those suppressed genes can restore that acetylation pattern and then generate more tumor suppressor and counterattack the tumors. And potentially these HDAC inhibitors can cure tumors. To summarize again, the acetylation effect is quite straightforward. More acetylation, more open form of chromatin, and that's a euchromatin, and then it is activated gene expression status. Reduced acetylation, they can induce into heterochromatin and then suppress those gene expressions. In comparison to the acetylations, other histone modification effect like methylation or phosphorylation is more complicated. It is not easy to say in one direction more methylation or more phosphorylation, whether it suppress or activate the gene is difficult to say. That is sequence specific and context dependent. If you want to do mapping of histone modification, how can you do it? Let's think how we did it for DNA methylation. DNA methylation case, it was only on cytosine residue and the information was only binary, whether methylated or unmethylated. And along the whole genomic sequence, we could make mapping whether each cytosine site was methylated or not. It's only one map we could do. Now in histone modification, There are multiple sites of modifications, and it can be either acetylation or methylation or phosphorylation or ubiquitination. So now it can be multidimensional informations. Along line of the genomic sequences, we need to draw the line for each individual modifications one by one. Maybe the information of a histone modification can be multidimensional and quite complicated. The approach can be quite similar to DNA methylation. For histone modification mapping, we use the technique so-called chip-on-chip. Here the first chip is chromatin immunoprecipitation. Chromatin is DNA structure. Immunoprecipitation sounds like still we need to use antibodies. Now we select one modification of a histone. That modification we call POI, point of interest. We choose one position of a histone and then one modification type, either acetylation or methylation. And assuming that we have an antibody for that specific position, the first step of this approach is induction of chemical cross-linking between histone and DNA sequences. After making this covalent binding, we can chop off those DNA sequences, and then some DNA sequences has histone, some is free, some are free DNAs. And utilizing this specific antibody for point of interest, and we can collect those histones. After releasing of these DNA sequences, and we can analyze those DNA sequences either by microarray or sequencing. If you use a DNA microarray, that is what you call another chip. This is a DNA chip. So then altogether this approach we call chip-on-chip. Utilizing DNA microarray, we can figure out which DNA is binding to these antibodies. Utilizing this DNA microarray data, now it's possible to mapping of those sequences along line the whole genomic sequences and figure out which area of DNA has a specific modification on those histones. If this second step of mapping is done by sequencing rather than DNA microarray, we call this approach as a chip-seq. In this case, direct sequencing fragment data can be utilized for the gene mapping, and again, along line to the whole genomic sequences, we can map out that specific histone modification happens where. By combining the two informations, one is the DNA methylation and histone modifications, these two are main regulating mechanism of epigenetics, and at least we can tell by looking at those chromatin structures. If they are highly condensed forms like a heterochromatin, this area is deactivated area, and if those gene area is open structures like a euchromatic structure, then this area should be activated area. At least two factors. If DNA is methylated, then this methylation can happen by DNA methyltransferases and also HDAC. HDAC is removing acetyl group from histone. If two enzymes act actively, then this area of genes are deactivated. So opposite way, if you use HDAC inhibitor or DNA methylase inhibitor, we can change the status from deactivated status into activated status. If you look at nuclear structures of a cell, usually outside of those nucleus, they have a lot of heterochromatin structures. Inside has a more dispersed euchromatin structure, but some area has a dark color. Those are also aggregated heterochromatin structures. If these heterochromatin structures are related to a disease, maybe we can send in HDAC inhibitor or DNA methyltransferase inhibitor, then activate this area of genes. Maybe we can provide new concept of drug development. And the huge size of this aggregation may imply that these genes are not gene-by-gene deactivations. This is a whole cluster of genes can be activated and deactivated together. Previously, we discussed about specific kinase inhibitors. In that case, we are talking about single specific enzyme inhibitor can be good drug candidates. But now, in these epigenetic approaches, rather than just a single enzyme, maybe it's kind of a cluster of genes here and there, and then we can turn on and turn off the whole area of those genes at the same time. If it is the right target, maybe this approach can be much more effective than just pinpoint out only a single enzyme target. There may be a controversial argument that whether this epigenetic approach can be specific or not. That's another important aspect that we need to think. But at least impact-wise, definitely epigenetic regulators can affect a lot bigger and broader impact it can provide to the cells.